So, welcome everyone to this sixth webinar of the WNT, which will be dedicated to sharing experiences with emerging technologies in other standard setting organizations. So just a little recap of the past webinars. We've had 4 of them, and I just added here the key messages of the first 4. The last one was a couple of weeks ago on identifying reference chemicals and building curated data sets. What are the approaches, issues, and learnings for the future? We had 2 speakers, and here are a few bullet points summarizing the key messages from the speakers. And of course, all of that you have available. You have the presentations of all these webinars. You have also the recordings as well as short reports. So here is the program for today. We will have 4 speakers, and we are starting today with Sarah Walton from the BSI group. Sarah Walton is the agri-food sector lead for BSI, the UK's national standards body. Sarah explores key challenges for sectors related to food, retail, and services alongside practitioners and experts to uncover real-world solutions. She has over 10 years experience of standardization development and related solutions. Sarah has led UK committees working in, amongst other subjects, food safety management, quality management, and occupational health and safety. Prior to working for BSI, Sarah worked on business-to-business information publishing, creating and delivering knowledge solutions for organizations based on their legal and compliance needs. So welcome, Sarah. Next speaker today will be Claudius Griesinger. Claudius is a neuroscientist by training with a PhD at Max Planck Institute for Developmental Biology on molecular mechanisms of the development of the visual cortex. Between 2006 and 2015, he worked at ECVAM, the Chemical Safety Unit, on stakeholder dialogue, ESAC coordination, and he was involved in the development of the test guideline 439 for skin irritation. Since 2015, he's responsible for activities on emerging health technologies and support to the implementation of the medical devices and in vitro diagnostic medical devices regulations in Europe. He's responsible for the establishment of EU expert panels on medical devices that has now been handed over to EMEA, the European Medicines Agency, in 2022 now. His current activities are on innovation in life and health sciences with a focus on artificial intelligence in health and innovative health technologies for cancer. So welcome, Claudius. Our next speaker is Monica Piergiovanni. Monica is a biomedical engineer working for the Joint Research Center of the European Commission. At the beginning of 2020, she joined the Unit for Chemical Safety and Alternative Methods that hosts EUREL-ECVAM, the European Union Reference Lab for Alternatives to Animal Testing. She's working on organ on a chip, focusing on standardization and regulatory acceptance with the goal to bring forward the field of these innovative, exciting technologies. She obtained a PhD in bioengineering at the Laboratory of Medical Structure Mechanics at Politecnico di Milano. Before joining the European Commission, she has worked on research and development of microfluidic devices for biological and medical applications. Welcome, Monica. And our fourth speaker for today is Rusty Thomas. Rusty directs the Center for Computational Toxicology and Exposure at the US EPA Office of Research and Development. His area of expertise is in the application of high throughput and high data content approaches to chemical toxicity testing. His academic training covers molecular biology and genomics. And following his academic training, Rusty performed bioinformatics and genomics research in the biotech sector and gained experience in high throughput screening and in vitro assay development in the biopharma sector. So welcome, Rusty. So as you know, this, as I said already, the webinar is recorded. It will be available on the WNT community site shortly. If you have questions to our speakers, please use the chat box for discussions and raising questions. Yes, let's remember the purpose of this series of webinars is really to collect food for thought for the December workshop discussions and to reflect on the challenges with the standardization, validation of new methods and emerging technologies. So on this URL, you will find all of the webinars I just mentioned, the recordings, presentations, short reports, and also I'd like to draw the attention of the members of the WNT who are present today, that on that same website you have the draft agenda for the workshop and also an issues paper that was distributed a bit more than a week ago with a request for responding with your initial feedback by 21st of November. So with that, I'll hand over to our first speaker, Sarah, today. Sarah, the floor is yours.
Hello, can you hear me?
Yes.
Excellent. So thank you very much for that introduction, Anne. I'm going to say a little bit today about the common standards system, the international common standards making system, BSI's role in that, and also give some examples of some areas where we've been working around new and emerging fields of work, including emerging technologies such as medical devices and AI. Just a few examples there. So next slide. There we go. So BSI is a national standards body with a national standards body in the UK, and we're one of a number of national standards bodies around the world. And this is essentially the international European standard setting system, the platform in which common standards, consensus standards are made and built. And BSI is a member of that. We were a founding member of ISO and IEC, and there's also obviously a Canadian standard body, French standard body. There are 167 members of ISO, just as an example of the number of bodies around the world. Sometimes those bodies are part of government, and other times they are not, as in our case, but we have a memorandum with our government, a memorandum of understanding to carry this role out for the UK and to make sure that there is a route into international standards making, as well as national, for UK interests and stakeholder experts. So the standards that are created, I mean, I think I've probably just covered that. International standards, regional standards such as END and CNs, and I think Monica will say a little more about that after in a bit later in this webinar. And obviously there are national standards at that NSB level, at the national standards level. The reason I wanted to show you this is because we also work on a sponsored standard at BSI, and other large standard bodies do a similar thing, where they work with stakeholders who have and were looking at innovative and new areas of work and want to bring something into market that isn't already a mature committee working on that field already. So we're doing a lot of exploration and research and trying to put things into the market earlier so that people could test them and understand and start thinking about the knowledge that is being disseminated about that new area of work and emerging tech. In a standards committee, a typical standards committee, we would want to make sure that we have the perspective and input from all aspects, all different representative constituent bodies to any particular subject or area of work or topic. And that would include government itself, regulators, academia, research bodies, standard users themselves, so that they can talk about how that works for them, industry bodies, professional institutions. These will always be representative bodies that we hopefully they would disseminate down into their own constituencies information about the work, the exploration that is being carried out at any level. And then from this standards committee, representatives would be nominated to attend international standard making committee work, so that way there is a throughput from experts from every country and from all of their aspects of particular subjects to go into that work. So an effective standard, well, obviously the legitimacy of these standards, they're very powerful tools. They can be all kinds of things, but they're essentially agreements. And the legitimacy of those agreements are around the fact that they are full consensus, they're open and transparent, they're voluntary in use. There is a public consultation around which has been discussed and delivered by the experts who have worked on and developed the standard. And they are systematically reviewed. So there is an iterative process I've shown in this diagram here. So an idea for a new area of work would come in and that would be assessed. And there would be a lot of research that would go on around that with the stakeholders. And that would lead to papers and thinking and a systematic process is into the next stage, which might well be, as it is here, shown the development of a standard, which is, as I said, completely iterative. There's always input and transparency and the opportunity for everyone to get involved, particularly at that public consultation phase. So different types of standards that you may be aware of. There are, when we're talking about the sorts of standards that maybe this group be aware of, maybe methods of test and very specific standards which would apply parameters. There may well be classifications or vocabulary. The things at the top of here, the ones I've noted here, the specifications, methods of test, vocabulary, classification. These are all very specific standards. They contain measurable clauses within them, and those measurable clauses can be the basis of schemes which are built by third-party certification bodies which are accredited, and those schemes can then be tested against and audited against by an independent third party, and they're part of that standards network, the quality infrastructure, if you like, which includes standard bodies making standards, accreditation bodies accrediting those things, accrediting certification bodies who are all independent and third party. You don't have to obviously develop something that is, you don't have to enter into that certification, but because it's all voluntary, but it is a very powerful tool for things such as tender documents. It's a language that people are speaking across the world. It's something very recognizable for people to understand. So you know things like BSI and ISO 9001 for quality management system, for example. And that is, it is a language people can speak to be able to work together. So in the case of innovation, I've referenced here some work that was done on the role of standardization in support of emerging technologies by the University of Cambridge, and that's where this diagram comes from. Essentially, in innovation, the earlier you get involved with standardization, the better, because it's about thinking systematically through the process as to what will help you, what pathways will take you to implementation and a possible scale up, understanding of new technologies in the market, but also ability to produce, the interoperability aspect, performance, safety aspect. All of these things can be considered very early on in the development of the actual innovation. And that is a way to help level the playing field, a way to help ensure clarity, simplify that complexity as much as possible, and also to start thinking about language in terms that everybody is using, so that, again, you're able to have the same conversations with people around the world about particular topics, product, systems. Also, you want to lay out characteristics and principles so that you allow the creativity to occur. And you're looking at output focus rather than thinking we need to deal with all of the very specific fine detail, to really think through how you're going to be able to open this for people to work together. So I just wanted to give you a couple of examples of how we've worked at BSI with other standards making organisations to think about a specific area which may well be of some relevance to what you're thinking about at the moment. So this is work on AI and medical devices, and I've referenced here my colleague Rob Turpin, who is the head of sector for healthcare at BSI, and could tell you some more about it if you would like to know more. But I can certainly speak to some of this work because I was involved in it earlier on. So essentially, what happened was we worked with the regulator in the UK, with MHRA, and big medical devices, the association in America for medical instrumentation, and also so BSI helped to bring together all of the experts from different countries, basically, to particularly in the US and the UK, to start thinking about how we would approach this issue of the emerging artificial intelligence and machine learning in healthcare. So there were many workshops for here, but there were some other, but there were 4 specific ones that took place, big stakeholder workshops, but many other conversations and meetings as well, to really start to think about with those experts what was going to be needed, what was really going to be required in order to support the development of this work, and also to provide reassurance to the market in every way that basically the regulations are being met, the safety aspects are being met, and performance aspects are being met, as well as the needs of the buyer, in this case, the NHS, UK, for example, or any healthcare organisation. The key thing about it was that really we were trying to look at how AI is different from traditional medical software, so that there was reference back to standards which already. So it was about really focusing on what was really going to be different in this space, and what the implications of those differences were going to be, rather than trying to reinvent any wheels around already existing standards that could be referenced as part of a navigation exercise when looking at this particular area of emerging technology. The MHRA in the UK published a software AI roadmap just recently, which you can find online on the MHRA, which really contains some very specific activities, which basically this has evolved out of these conversations over the last few years. These are things that we would like to go on to do next, because we feel it will support the next stage of what we need to be thinking about in this whole space. So these standards were of particular interest in with 2 routes of standards, which I've already sort of mentioned, where we're thinking about guidance to align global medical devices to global regulatory standards on the risk aspects and the approach that needs to be taken in order to meet those regulatory aspects, and also best practices around what needs to happen for safety purposes and criteria for the adoption of that AI in healthcare environments. So two different standards. These are about to appear. One of them is at, I believe it's the British standard at the moment, which will eventually become proposed further into international state. Is about to be published, and the other standard is at the DIS stage, which is the draft in international states, where they're thinking about, and that's got another few weeks to run. So if you want to have a look at that and make some comments on it, you're very welcome to do so. So there are two there. I can be clear about that when we can look at my notes, but that's something to be aware of. I won't say very much about this because this is just some information about the focus group that's happening at SENLEC about organ on chip. But I think Monica will cover that in much more detail. But essentially, this is about the focus group. They're doing a very similar thing from, as I've just described, happening around medical devices and AI, where there's a lot of research and thinking going on at an early stage, systematic thinking, exploratory thinking, in order to really decide with the experts what the shape of the future will look like, so that we're not instantly thinking about the standards, but thinking about how we get to that and what will be required to help support and give infrastructure to the emerging technologies and industry there. So you can see the working groups which have been set up. There is a roadmap which is due to be published or completed next year, which will do a very similar thing to the roadmap that I was there with around medical devices and AI. Just very quickly, industrial biotechnology is an area of work, again, which is a big piece of research with the Industrial Biotechnology Leadership Forum and with the UK Research Institution, part of government. And we facilitated a lot of the experts to come together to think again, to do some thinking around 5 different sectors: agri-tech, biofuels, fine and specialty chemicals, plastics and textiles. And we came up with some recommendations, pathways around selective resources, communication, proper approaches to these things. And I've given you a link to the report if you're interested in what happened with that and where we're going next with that piece of work. Again, this will be supporting innovation with standards. One piece of work that came out of that, one strand of new research, was work on cultivated meat industry, where we're thinking about a lot of ethical issues there, a lot of health and safety issues, which are very similar to things that happen in the pharmaceutical industry, of course, because we're thinking about both serum being used in bioreactors and how that and what the implications of that are, how to make sure that is the right grade and safety to provide reassurance for the market in the future, so there can be investment and scale up as necessary. And that report, there's the first white paper will be coming out, which will be available to everybody on our website, which will be coming out probably early next year now. And I've put some more links here for you to look at as well. Hopefully that will be of some interest to you on both on the ISO site and also a few things around standard innovation on BSI. I hope that was helpful. Thank you very much for your attention, and I'd like to my email address as well as my colleague.
Thank you very much, Sarah, for sharing all of this knowledge and also some useful links that I copied in the chat box for participants if they want to have a look, of course. And feel free to, I'm talking to all participants, if you have any questions to Sarah, to use the chat box. And now we are going to move to our next speaker, Claudius. Are you ready?
Yes,
yeah.
Let's see. Okay. So, I hope you can see my screen and you can hear me well.
Yes, very well.
Okay, good. So, good afternoon, everybody. Thanks a lot for this opportunity to present some insights maybe about the use of standards for product conformity assessment in the area of medical technology. There's a tiny little bit of overlap with what Sarah just told you, but it's actually not too bad. So that's good. I'm trying to cover 5 points here. First, going to talk about product conformity assessment and use of standards, life cycle and risks, the modular use of these standards, very briefly on emerging technologies, AI and health, which is a focused interest of us, and then provide maybe some summary reflections if we have the time. So standards already has been set. I mean, maybe what's interesting here is that there are also other standardization publications, such as technical reports, that are also actually quite important sometimes, and they are all voluntary guidelines. Voluntary, it's important, providing the technical specifications for product, services, and processes. Sarah has showed you already very nicely standardization organizations, so I'm quick here. What I would like to tell you about the product legislation in the EU is we had in the EU basically the so-called old approach, now old, until 1985 here on the left. And that meant actually that in about 700 directives for many different products, you had very detailed technical and administrative requirements spelled out in these legislations. And of course, that collided a bit with the rate of innovation that, that you would have to update basic legislations when there are changes in innovative technologies. And also there was a realization that there are often common risks. So there is a possibility to describe and deal with common requirements, basically, which led then to the introduction of the so-called new approach in 1985, which means, and that's essential, that the legislations contain only essential requirements and the technical details are EU harmonized standards. I'm coming to in a second what this means. It also meant that Europe then developed, or the EU developed, a standardization policy and the instruments for conformity assessment, which means the product evaluation against standards. That was all then solidified, so to speak, in the new legislative framework. I'm not going to talk about this. You can look that up if you're interested. But important is this rate of innovations that triggered also this new approach with the use of standards. And here I'm just showing you a slide that is quite nice. It shows the evolution of the pacemaker from 1958, the external pacemaker, until 2016, quite recent, with the intracardiac pacemaker. So there's a lot of innovation going on in medical technology and medical devices. And we're still having that problem. We just, in about 2 years ago, developed the guidance documents for expert panels on evaluating novelty of devices, which is an important aspect in the regulatory approach to medical device safety assessment and clinical evaluation in Europe. So it's also not that easy, actually, always to say what's really novel and innovative. Is it an incremental innovation? Is it a disruptive, really completely changing innovation? So on the medical devices, as I said, they are basically considered products in the EU. So it's a bit different in other jurisdictions. And so they go through a conformity assessment by the manufacturer and notified bodies where required. That's different for other health technologies like medicinal products, drugs, or vaccines where we have an authorization scheme. I just wanted to highlight that. Nevertheless, the assessment principles are often quite similar, and the life cycle approach is similar, the risk approach. And of course, we also have a lot of combination products where you have medical devices coming together with drugs, actually. So the way the medical devices are regulated in the EU is through this new approach, I said already, to technical harmonization and standards. That means there needs to be a close relationship between EU law and standardization requests by the Commission. So the Commission gives mandates to European standardization organizations. And if the product is manufactured and over the life cycle, basically, and managed in alignment with the harmonized standards, then it is presumed to be in conformity with essential requirements. What is important is that this system has also some limitations, and they came up, and you will have now in a lot of product legislations, also in the upcoming EU AI Act, we have provision for common specifications in the medical device legislation as well. That means if there are no harmonized standards or they are considered insufficient, the Commission can actually issue so-called common specifications in lieu of a standard. And the expert panels, I will talk a little bit more in a moment, have a role for these as well. So very briefly to sum that up, conformity assessment is against harmonized standards in Europe, and that means basically that if the notified body or the manufacturer look at the harmonized standards to confirm the conformity basically of the device with the standards, then you have directly a conformity with the essential requirements of the legislation. So it's a quite elegant system and has reduced the number of legislations actually quite a lot and simplified them considerably. There is also an element next to the notified bodies of expert panels, as I said, and the competent authorities in Europe that have a scrutiny role. It's also important to keep that in mind. So the harmonized standards basically are standards that are developed on request of the Commission by the standardization organizations, and they basically offer a guaranteed level of protection for this conformity assessment. But it's also important, of course, to realize that industry notified bodies may also use non-harmonized standards for evaluation. It's just you cannot do really a conformity assessment in Europe with this. The important is also that the application of standards is voluntary, so they are not legal documents. It's not like the test guidelines in the EU test method regulation, for instance. And a manufacturer may also decide that he's going or she's going for another technical solutions. In that case, the manufacturer, though, has to demonstrate that these are answering the needs of the essential requirements, which again would involve typically a notified body. So I wanted to talk a little bit about expert panels in medical devices, conformity assessment with standards also. We have a lot of innovation in that field. A lot of the devices that are very innovative are, of course, high-risk devices. And you need also there, of course, good clinical evidence and a robust risk-benefit determination. And of course, what's very important, adequate post-market clinical follow-up. What I wanted to emphasize also is that the legislation that we have in Europe basically realized in a way that the notified bodies do not always have the necessary clinical expertise, sorry, it should be called evidence, but expertise. And also the competent authorities cannot be specialists in all relevant clinical and scientific fields. Therefore, there is this new element of expert panels, which sits in a way between the notified bodies and the competent authorities, with advising the notified bodies on specific questions for high-risk devices and also providing input on potentially on common specifications. Just very briefly, these expert panels we set up and handed them over in 2022 was a quite complex landscape. Again, they help with the consultation and contribute. So it's important to realize that the notified bodies also have problems, for instance, to recruit really clinicians that can really go into the depth of a clinical evaluation dossier for medical devices. And that was the solution then to have these expert panels. It's clear that this is a risk-based approach. I'm not going into depth in this. We have class 1 to class 3 in Europe. The market share is very illustrative, and you can see class 1 would be stethoscopes or scalpels. Class 3, for instance, vascular stents would be the high-risk things, surgical meshes, artificial hearts, etc. Also important is that the standards that are being used for conformity assessment and indeed for assessment for conception, design, and so on over the whole life cycle, they play a role over this entire life cycle. They're not just important for the conformity assessment, they're also important for all the other steps that you can see here, from concept to planning to design verification, validation with the preclinical testing, clinical investigations, etc., over launch to market. And also what I would like to emphasize is that clinical evaluation is spanning not just the post-market, the clinical evaluation time, but also goes basically into the post-market phase and the post-market space. There's a constant obligation of the manufacturers to collect information on their devices, how they're basically doing on the market. And also there are standards for doing this. Now, in a way, what is maybe closest to the WNG, I thought, was tests for biological evaluation, for instance, so toxicity, also genotoxicity, irritation, etc. And you see here, this is just a part of the ISO standard 10993 on biological evaluation. There is also, that's interesting, maybe, there is an example at least that the TG 439 basically was fed into the ISO standard. Actually, so it was first a test guideline for skin irritation that then became an ISO standard with, of course, a different SOP. But I think that example here is most close to what you're really discussing, not standards on quality management systems and so on. On that, I'm leading to the next point on the modularity. If you consider the example of a cardiac resynchronization therapy device / defibrillator, that's basically a pacemaker. And these pacemakers normally nowadays are also implanted defibrillators. What you can see here is all the different standards, not all, some of the relevant standards, there are many more, of course, that you could use for that evaluation, over particular requirements of the safety of such defibrillators, electromagnetic compatibility, the 4-pole connector systems, the dimensions, symbols to be used, particular requirements for active implantable medical devices, and then the very basic ones, in a way, very important ones, the risk management system for medical devices and the quality management system for regulatory purposes. That are very horizontal standards. That just gives you a flavor a bit how over this life cycle you apply many, many different standards, also for the development when you have an idea for a new device, but really also up into the post-market space to make sure that the device is safe and that you're vigilant about potential adverse events that could have happened. Very briefly on the artificial intelligence, that's of course a complete game changer for many sectors, but also for healthcare. And so that's an interesting example where now standards will play a role, of course, and also harmonized standards done in the EU. There are already some horizontal standards that in a way cover what we call in the EU, but also I think in the US, the term is used, trustworthiness, different modules in there. So you see some of these standards, but of course it remains to be seen how useful these standards are for specific sectorial needs. Just highlighted here, healthcare is one of the sectors really where a lot of earnings are already created, at least in 2018 to 2019. Finance is at the top, but health technology is faring very well. And there's a plethora of new developments. It's quite fascinating. So the question will be really then to see also how these horizontal standards are sufficient, or whether there is sector-specific guidance needed, or standards for health, for finance, for transport, and education. And that gives you maybe a bit of a flavor also of how this system in a way works, irrespective on an international or European level. It's always a bit of a game also to see, you know, are you still up to date? Is it fully applicable? Are there specific questions in a sector that maybe are not fully dealt with in a specific more horizontal standard? Let's say, for instance, data quality, where you have many specific issues about health data that you need to use basically to train a system. Okay, and then I want to close first with a very simple overview table. I tried to compare a little bit the standards with the OECD test guidelines, how I understand them. It's a while ago that I was more involved in the field, and probably I'm not up to date with many discussions that you had in the meantime. The scope, first of all, is very broad for standards. It's technical specifications for product assessment. It seems to me it's more focused for the test guidelines, with providing basically scientific methods for testing materials' properties. Also for making predictions, so there's a diagnostic element often in there, and for risk classification and labeling. Use for risk classification, it's in the medical sector in the EU, not that critical, I would say. You could say Rule 19 and the MDR about nanomaterials, maybe there are some standards could help with defining whether the rule applies or not, but generally it's more about the use scenarios and the technologies. Whereas of course the test guidelines are playing a major role for risk determination, risk classification also, and labeling. Now, on the regulatory use, the standards are voluntary. That's important. But of course, if they're used, especially the harmonized ones, they really help you very much with gaining conformity presumption. The international standards have a high regulatory interoperability. I think Sarah alluded to this already. And of course, the OECD test guidelines we have in the EU, there's a legal status, they go into test method regulation. And of course, there's a high regulatory interoperability because of the MAD. The issue, it's standardization bodies. So it's not notified bodies, that's very important. They may assess non-standard-based solution by a manufacturer, but they're not really typically that involved in standardization from our understanding. The OECD test guideline program, of course, for the test guidelines. The supervision is notified bodies where required. It also could be a self-declaration by the manufacturer. And then higher level supervision by the competent authorities and agencies. This is equivalent here. And the bottlenecks, of course, standardization is slow. It's not a fast process at all. Also, international standardization is not fast. For the EU, the harmonization requests are not always taken up. So that led also to the model of common specifications that I told you, which we have actually only at the moment for in vitro diagnostic devices. And there's not always a full competence of notified bodies. And it's not a reproach, and don't get me wrong, it's just very difficult also for notified bodies to recruit, as I said, experts on very deep specific clinical competences, for instance, and therefore we have this element of expert panels. Similar validation regulatory acceptance can be slow. Now I'm closing with the last slide, and probably I'm telling you lots of things that you've heard already, so bear with me. So Anne asked us she gave us 3 charge questions, I would say. One was TGs are based often on one specific assay. So, think about that. What about transferability? What about review mechanisms? And I have put ample question marks on this slide because I think probably the discussion has moved on a lot, but I was again thinking actually over the weekend a bit. It seems to me the problem obviously is that the rate of technological progress is high. You have a lot of very sophisticated mechanism-based assays coming up, and validation and regulatory acceptance, on the other hand, are often relatively slow. So what are avenues for solving this or for addressing this? I mean, maybe it's a mix of a process renewal and also paradigm renewal, and probably there you had lots of discussions. I really think that validation, in a way, the model needs to be rethought, but that's probably underway. And also maybe the test method paradigm. So it seems to me, perhaps the test methods are still too much predictive machines, providing directly, trying to model the regulatory required information, and not so much the emphasis on the scientific methodology that you use with many other methodologies to come to a conclusion on a specific question. But maybe I'm wrong. Also, I think there always has been the question, do we try, or you, I'm not so much active in the field anymore, to fit new technologies in the mold of traditional technologies? Do we reverse engineer too much, maybe, instead of looking what is in the biorevolution happening on picking up mechanisms of action that are relevant for toxicity? I also wanted to finalize to just say that the expert panels maybe is an idea that is interesting for everybody to think about as a way of a mechanism for reviewing, because you get really top-notch experts there that can help you. It's difficult to be really behind all sorts of new technologies and knowing in depth. So I think getting external expertise from high-level experts is a good mechanism. It costs, of course, money as well, but it saves maybe some time. And the last, maybe thinking about all this together, process renewal with a more flexible and modular use of test methods, including within integrated and assessment and testing approaches. Maybe more weight of evidence. And with that, I thank you very much. I hope I'm a bit more or less in time. Thank you very much.
Thank you very much, Claudius, for this very insightful presentation and for sharing your reflections. I think that's very, very useful. And if people want to react in the chat box, please don't be shy. And without further ado, we'll move to our next speaker, Monica. Ready?
Yes, let me share.
Very well. So
not this one, but maybe this one.
Yes.
Okay. So thank you, Anne, and yeah, good afternoon, everybody. So my name is Monica Piergiovanni. I'm here to talk to you about the role of standards in innovation, and specifically I am tackling the case of organ on chip. So I imagine that all of you have a rough idea of what an organ on chip is, but just to be on the safe side and to be on the same page, we can define organ on chip as fit for purpose devices that contain one or more engineered organs, organ substructures, or functional units. And these are cultured in a controllable microenvironment. And really the final goal of the organ on chip is to represent one, let's say, functional aspects of a specific organ. And organ on chip now come in a huge variety, I would say, of designs and purposes. So here in the picture, I reported some of the most common devices. So we have, of course, single organ systems. We have multi-organs that connect two or more organs together. And finally, we have the final ambition of having a human body on a chip. But we have to consider so that if we want to tackle the organ on chip completely, we have to take into consideration that the biology is very complex. So we want to have 3D models and also to combine them together. But we also need to consider that we have different engineering aspects, mathematical aspects that now come into the device and have a higher role. And this is where we want to understand if standards can have a role. So I will not repeat the definition of standardization because I think that now you get it, but I would like to underline the role of standards in the innovation and in particular in the technological innovation. So standards are usually developed to help companies or manufacturers to demonstrate to the customers that their products has the feature that they claim to have. And this is the first, let's say, role of standards that I see. But the second one is also to ensure that we have an acceptable risk for health, safety, and the environment. Of course, there are many other benefits of the standardization process in innovation. So, for instance, it's a way where we can innovate more, and we can have a stakeholder group that are sharing also knowledge. And also facilitating the processes and the integration in the value chain. But of course, every innovation has to pass through, let's say, specific technology readiness level. You may know the TRLs. And this slide, which I took from our colleagues in Sensenelec, is basically showing you that from the very beginning of the knowledge development, so when we are in the very basics, in the very low TRL levels, it's necessary to have an understanding of the terminology and on the language. So semantic standards in the early phase are very fundamentally necessary, let's say, for development. But when we go on with the technology development and we start, applying it and testing the innovation into the lab, the role of measurement standards increases in importance. And we can say that we also have a growing role of interface standards when we want to ensure the interoperability of our let's say innovation into the whole system, let's say in the lab, or if we want to integrate components from various sides. And finally, when we are in the, let's say, higher levels of readiness, we are, of course, trying to set up our business and enter into a common use. We have a higher role of, sorry, of quality and safety standards. And I would like to give you just a couple of examples to make you understand exactly what I mean. So here I picked the case of the multi-wells that we all consider to be commodities in our lab. But of course, these were at some point in time, these were considered innovation. And this point in time, by the way, was an influenza epidemic in the 50s. So we have a Hungarian medical doctor that started to develop the concepts of multi-wells. And in time, these devices were at various shapes, various materials, and every company has their own. So this means that if you want to use or perform a specific assay, you should have the specific, let's say, device and its own multi-well together. But only with in the early 2000s, I would say, when we started to use the fluidic handlers, at that point, the role of having an interface standard was most relevant, because we wanted to be able to buy multi-wells from different vendors, but being used with the same robotic platform. And this is where a series of standards was published by ANSI to ensure that the geometry and the depth of the multi-wells were conform to the specifics. And then another example that goes more on the quality standards, but is still tackling an innovation. So if we go on most recent times, and I picked the case of genomic sequencing for diagnostics purposes. So we can say that even if the technology is quite established now, we have high throughput DNA sequencing. But however, if we want to use these techniques in the clinical settings, for instance, we have to ensure that the quality of the results is maintained. And in this case here, I reported these 3 standards that are some example of how a standard can guide us and suggest the correct metrics to control the process in all the steps. So from the sample and library preparation through the sequencing process itself, and finally from the data management. And please consider that this is tackling a huge, not huge, a big, let's say, a big number of different technologies from different vendors. So I, of course, try to give you some example of standardization, let's say, per se, of standardization from ISO and Sensenelec. But of course, if we think about the world that we work, there are many other standards around us, either guidelines or guidance documents that you know much better than I do, probably. But with this slide, I would like to be on the same page with you on using the word standard to incorporate all of these, let's say, different documents and different settings that we are talking about. And to go, to start slowly going into the organ-on-chip case study, I would like to tell you that the way that we want to see standards in this context is to demonstrate the technological and biological relevance in order to increase the implementation by the industry and provide the basis for regulatory acceptance. So this is, let's say, the framework where we were working when we picked the case study of organ-on-chip. And as a first step, last year, we started to investigate the role of standards in this broad sense. And you see here the publication that came out where we performed what it's traditionally, let's say, called pre-normative research. So an analysis of all the standardization activities and documents present in the literature. So I encourage you to read it, to have an idea of what this means and which are the aspects that can be tackled by the standards. So we covered various aspects. For instance, we covered aspects like the properties of the material used, the assessment of the performance, some quality criteria, and so on. And as an annex to the publication, we also compiled a database of published standards that were developed in different fields, but that have some aspects that can be related to organ-on-chip devices. And you have, of course, the list, the title, and where we saw the commonalities with organ-on-chip. And following up on this analysis, we tried to go a step further. So we gathered together, I think for one of the first times, standardizers, developers, and regulators in the same, let's say, area, the same virtual workshop at the time. And so we set up this putting science into standards workshop that, of course, can also be a putting standards into science workshop, depending on your perspective. And we tried to pick up the discussion and identify some pillars where standardization can have a role. And I will now like to give you a hint of some of the aspects that were discussed and that I think are more, let's say, relevant for this case study. So the first one is the performance. So if we want to have a credible organ-on-chip device for toxicology, but also for biomedical research purposes, we have to trust and believe the engineering aspects. So we have to trust that the material is defined and that the surface treatments that are sometimes useful are compatible with the biological problem that we are tackling. And with the organ-on-chip devices, there is often the issue of the adsorption of small molecules into the plastic. And of course, this is something that can be, I would say, quite easily tackled by a testing standard of or a measurement standards that allow us to really evaluate these phenomena rather than simply accounting for it. And another also aspects that is unique of organ-on-chip devices is the presence of sensors and of fluid flow. So of course we want to have sensors and actuators in order to be able to understand the electromechanical action of the cells, try to, for instance, stretch them if we are talking about a lung on a chip, or if we want to be able to measure the electric conductivity of a cardiomyocyte tissue, for instance. And of course, this sensor needs to be accurately calibrated and maintained also in time. And a similar consideration can be done for fluid flow. So in the case of the organ-on-chips where we have let's say, two organs that are connected by a fluid flow that's supposedly to represent the blood flow in our body. It's of course necessary that we are able to measure the fluid flow and maintain it in time and be sure that what we think we are applying with our pump is actually what the cells are sensing at the microfluidic level. A second, let's say, area where standards can facilitate the use of organ-on-chip, I would say, is usability, which is a bit similar to the interoperability and interface standards that I was talking about. And clearly, we have to make sure that the fluidic connections and the control devices are accurately designed and are correctly, let's say, operating, but also compatibility with lab equipment is also key, especially if we want to make these devices compatible with our robotic platform. Of course, we have to comply with the standards that we saw just before, and we have to make sure that all the geometrical tolerance of the device is maintained. Otherwise, we will break everything up. Also reporting, I would say, is another pillar of standardization. This is mostly related, I would say, in the standardization area to clinical application when we want to correctly report data from patients, but also in the chemical domains that's relevant. And I would say that for organ on chip, we have to sort of consider which are the specific features of the organ on chip that we want to report. So of course we have to let's say extend a bit the classical reporting standards that we have in mind to also include those parameters that are more let's say hidden below. So for instance, I mean the fluid flow, as I mentioned, or the calibration of the sensor. So all these very technical and very specific parameters should also be reported in order to guarantee a transferability and a repeatability of the results. Finally, yes, just a note of I mean, maybe it seems obvious, but let's always remember that organ-on-chip devices are very complex multi-wells. So basically speaking, they are plastic labware in a very complicated way. So it's important that when we operate those and we insert the biological model inside the organ on chip, it's of course important that we follow all the guidance documents and the good practices that we have, because in the end, it's not really something totally different from complex in vitro methods, let's say. So if we want to know more, and also all the information that I didn't have time to include today, please have a look at these two documents. So one is a peer review paper and one is a workshop report where we have summarized all the outcome of the workshop. And so to conclude, as Sarah was already spoiling at the beginning, we had established as a follow-up of the workshop this Sensenelec focus group that is chaired by the Netherlands, and it's now, let's say, starting its operation since 5-6 months, I would say. And I would like to invite you to join the group, especially to join the working group 5, where we are planning and we are trying to understand perspective from regulators and from end users that we can consider to, let's say, define a priority roadmap of the standardization needs in the organ on chip. And yeah, with that, I would like to thank you. I'm sorry that someone entered. I put a note, but it was in English, so probably not the best choice. So I'm sorry for the interruption and thanks for your time.
Thank you very much, Monica. No problem. You can stop sharing if you want. Yeah, thanks. Very insightful again, and yeah, lots of resources for us to consult on. Without further ado, Rusty, please go ahead. Yeah, you are muted.
Yep, I got it. Let me share here. I'm hoping nobody enters my room, but I have 2 kids getting ready for school this morning, so that's certainly a possibility on my side. So all right, I need to swap, right?
You are on presenter mode, so you need to swap.
There you go. Are we good?
Yeah.
All right, so I'm going to be something completely different. But thank you for your attention and inviting me. I'm going to have a wildly different presentation probably than the previous few speakers, but and I'm going to try to do it relatively quickly, but give you some idea of what I see as the keys to development and application of emerging science through the eyes of a researcher in a center that's tasked with that within a regulatory agency. So I'm going to frame real quickly frame my the concept of how I'm approaching emerging application and development of emerging technologies into a regulatory organization by the original paradox. And just quickly, if you're not familiar with this paradox, according to legend, that original Greek paradox was centered around a Tumessian fox, which was big and destined never to be caught. It was said that the fox was sent to the gods to prey on the children of Thebes as a punishment for a national crime. But in order to protect that children, a Theban general discovered an ingenious solution to this problem by fetching a magical dog, Laelabs, who was destined to catch everything it chased. Since Zeus was faced with this inevitable contradiction, he solved the problem by turning the two beasts into stone, and the pair were cast into the stars and remain as Canis Major and Minor. I kind of view that regulatory application of emerging science within that same lens, that it's also a paradox, right? That we have chemical hazard and risk evaluations having similar contradictions. There's a widespread desire to use new science to test chemicals and make decisions in a more efficient and greater human relevance. But at the same time, we have an entrenched toxicity testing system and regulatory process that's difficult to change and evolves slower than the science and technology is evolving, right? So how do we solve this problem? Well, despite the title of the talk, I'm not necessarily claiming to be Zeus, but I'm going to say that I have, what I'm going to try to provide is at least my perspective on a handful of these key principles or considerations that may help in the development of application of this emerging science to chemical safety. And I'm going to try to walk through these very quickly one by one. And I have 7 of these keys. So I'm going to shift. I won't go over them here, but I'll go over them in the next 7 slides. Promise 7 slides, 1 for each key. All right, the first key, as I see it, is to accept that most environmental and industrial chemicals interact with biological systems in a non-selective manner. In other words, most of these chemicals are very promiscuous in the biological processes and targets they perturb. On the left-hand side, you see a cumulative distribution plot that is one way to look at this principle. Right, the data are industrial environmental chemicals in our ToxCast library, and those are evaluated across a broad suite of ToxCast assays that evaluate a broad range of biological process and targets. Right, the x-axis is the concentration that is required to activate the first assay, divided by the concentration to activate 10% of these biological processes and targets. For most of the chemicals, you see that there's a less than a 3-fold difference between the two. The lesson here is that no matter how good your technology is to identifying a predominant mechanism of action for a chemical, most of the time this is likely to be impractical. This is similar to established concepts in ecotoxicology with specific acting and non-specific acting classes, and where they have also observed that the majority of industrial environmental chemicals are that non-specific acting type. And overall, what this suggests is that bioactivity, in vivo or in vitro, can be a pretty good surrogate for potential adverse effects in chemical assessments. The second key is to develop and apply toxicity testings and integrated approaches that contain emerging science with a general lack of chemical selectivity in mind. For many of the next generation based approaches that have either been published by my group or as well as other groups, predominantly in the cosmetic product related research literature, right? They kind of fit into this general framework on the left. One is through the use and application of broad coverage technologies and models, such as transcriptomics or high content imaging systems that evaluate a broad landscape of biological targets. And then you also couple that with target-specific technologies and models in some sort of integrated combination, similar to an IATA, that looks both broadly at biological space and also hones in on those important receptors, enzymes, and other biological processes that can be frequent targets of industrial environmental chemicals. And then combining that information in a way that sort of differentiates between these non-selective modes of action as well as those selective modes of action in some sort of distinguishing way that allows you to either perform a bioactivity-based assessment or more of a mechanistic AOP-based assessment process. The third key, in my opinion, is to distinguish between protection and prediction in both existing and new technology approaches, right? On the left-hand side, you see that there are two studies that were done in the pharmaceutical industry that were comparing toxicological results from preclinical animal studies from those in early phase clinical trials. I highlighted a couple key takeaways from the abstracts in these studies, right? In the first study, the results showed that true positive human toxicity concordance rate was as high as 70% when you looked across all preclinical species. But when you looked at only rodent species, the true positive concordance rate dropped to around 40%, right? The overall conclusions were similar in a second study with a relatively low positive predictive value for many species and organs, but with the negative predictive value was much stronger performance measure, indicating that an absence of a toxicity in an animal study more strongly predicts a similar outcome in humans. This suggests, at least in my view, that animal studies are not necessarily predictive of the specific hazard outcomes in humans, but when used collectively, they are generally protective, right? The new science and technologies can be applied in a very similar protective manner. On the right-hand side are two case studies where in vitro bioactivity, as evaluated across multiple suites of in vitro assays or using some of the emerging science, that when you convert these general bioactivity using a administered dose equivalent or in vitro in vivo extrapolation of pharmacokinetics, you actually observe to be protective of a toxicological response in vivo rodent studies. The top graph is a case study that evaluates that principle using a ToxCast battery as the broad suite of in vitro assays, right? And this case study was performed within the Accelerating Progress in Chemical Risk Assessment. The bottom set of assays was, the bottom set of graph is a similar evaluation using high content imaging as the measure of biological activity. And the results of that case study were generally the same in that in vitro bioactivity was a protective surrogate for adverse responses in vivo toxicological models. Generally, the response in these in vitro assays, although conservative, weren't as conservative as other measures such as the threshold for toxicological concern. The fourth key to the development and application of emerging science to toxicity testing is to extend the definition of hazard either through an integrated measure of bioactivity, as outlined in that previous slide, for the protective application, or to use specific assay responses in an AOP MOA context to be predictive of the toxicological responses. You can see that bioactivity-based assessment more aligns with our protective approaches for those non-selective chemicals, while for those selective chemicals, you can focus on an AOP MOA-based application to be predictive. And on the bottom right-hand side, you see that an example of that are target-specific assays for TSHR inhibition or TPO inhibition and NIS inhibition are key events in thyroid toxicity, and trying to integrate those in an AOP context to be predictive of thyroid-related responses. The fifth key is to characterize the variability in performance of our existing and new toxicity testing methods. I think we've done this for many of our new methods using the emerging science, but we've not been quite as diligent on characterizing the variability in performance of our existing methods and models. A couple of exceptions have been for acute toxicity and skin sensitization, but we've really yet to do this for many of our repeat dose toxicity models, right? Part of this is because we typically don't repeat these studies multiple times due to cost and time requirements. However, below are a couple studies of our group attempting to do this. Katie Paul Friedman and colleagues have been using a highly curated database called Tox4FDB that contains about 6,000 studies on 1,100 chemicals. We've been analyzing this data in different ways to get at the question of qualitative and quantitative variability. Although there are limited examples where that higher tier toxicity studies are repeated under exact conditions, such as dose, route, strain, et cetera, one can look across those repeated and those repeated under similar parameters to ask these questions. On the left-hand side, Katie and colleagues analyzed the reproducibility of low observable adverse effect levels using study parameters such as route, strain, et cetera, as covariates. While in another approach, they filtered the data down to look at reproducibility of these low AL values only in highly similar study designs. She also broke out subchronic from chronic studies to see if study duration had an impact. But the take-home message is that no matter how the data were analyzed or the duration of the studies, the quantitative variability was generally within a root mean squared error of about 0.4 to 0.6. What does this mean? Well, a root mean squared error of about 0.6, the 95% prediction interval of a low AL value was plus or minus tenfold, meaning that if you repeated that subchronic or chronic study more than once, the low AL value probably would be within that tenfold, plus or minus tenfold range. On the right-hand side, Katie and colleagues evaluated the qualitative concordance of the target organ effects while breaking down that concordance based on sample size, study type, species, and target organ effects. And the qualitative concordance ranged from about 40% to 80%, but was generally within a 60% concordance rate. So you can see how reproducibility from a qualitative standpoint, these repeat dose studies were. The sixth key to the development and application of emerging science, in my opinion, is to evolve the validation or qualification process to be more fit for purpose and performance-based. I think some of this was touched upon by the previous speakers. While traditional toxicity testing methods have not changed very quickly, but I noted at the beginning, the advances in science and technology for toxicity testing are occurring at a much faster rate than our current validation process and can keep up. And although general OECD guidance on validation states the validation process to be flexible and adaptable, our current application of this guidance is generally focused on a one-for-one replacement of a reference method using a ring trial design that takes multiple years and significant resources. To evolve the process, different expert bodies and organizations have been proposing a shift to a more fit-for-purpose and performance-based process. This is reflected, for example, in the 2017 report on using 21st century science to improve risk evaluations. In that report, the U.S. National Academies emphasized the importance of defining purpose and scope of the new technology. It also recognizes challenges in validating a new technology where there's no gold standard or against assays necessarily that have not been themselves validated. It also suggested establishing performance standards for data quality and recognized that a ring trial design is not necessary for all purposes. It also emphasized the need for reporting standards and transparency. On the right-hand side, you see that there's a publication that evolves these main concepts further and puts forth a general framework for establishing scientific confidence in these emerging scientific methods. You can see the main components of this framework include fit for purpose, technical characterization, biological relevance, independent review, and data integrity and transparency. Very similar to what the NAS recommended. Lastly, the seventh key to the development and application of emerging science to toxicity testing may be to realize that fundamentally the new paradigm that I touched upon may not be that different from our old paradigm after all. On the left-hand side is a general representation of our current in vivo toxicity testing paradigm. If you accept the results of the preclinical to clinical toxicity studies, you can say that many of these broad coverage toxicity tests, such as the repeat dose subchronic studies and chronic studies, right, that use apical endpoints as a definition of hazard, are generally protective, while the hazards observed in those apical-based tests aren't necessarily observed frequently in humans. However, the mechanism-specific tests, those that are designed to specifically evaluate particular mechanisms such as receptor activation or pathway inhibition of an important toxicity pathway, if they are human relevant, and evaluated for human relevance using an AOP or MOA framework, they can be predictive. On the right-hand side is the parallel paradigm for using new and emerging science. You can see that they are integrated when integrated in appropriate ways, you can achieve the same general testing and assessment goal of human protection for the non-selective chemicals and prediction of potential adverse effects for specific acting chemicals. I guess as the old proverb says, there's truly nothing novel in existence, and every idea has a precedent or an echo from the past. So I think what I tried to touch upon was these 7 keys, at least from my perspective, of moving from a paradox of that emerging science evolving faster than our regulatory paradigms can accept, to maybe a new paradigm of more quicker adoption of these new technologies and approaches. And how do we approach that? And that, I think I've out of time, and I'm happy to touch upon any questions in any time that we have. Anne.
Thank you very much, Rusty, for this very interesting presentation. There's lots of material that has been, lots of ideas presented today. We don't have any questions in the chat box for any of the speakers today. So either we had a very shy audience, or the participants were following with a high degree of attention each of the presentations. In any case, the webinar is recorded, so people will have the leisure to watch again the video and go over each of the presentations. And if there are any questions, you have also the contact details of our speakers. So I would like to thank you all, 4 of you, for very insightful presentations. I think that was the webinar that was maybe the most out of the box thinking for the WNT. And we really wanted to have this opportunity of looking at what's going on in other organizations, standard setting organizations, or or agencies. And I think that will be a very useful material we can take forward to our December workshop. We are finalizing the program and materials. So thank you again, and I wish everyone a very nice day. And we'll be in touch in future, I hope, at least. Thank you. Bye-bye.
Thank you. Bye.
Thank you. Bye-bye.